You may consider it a systematic knowledge or a practice or a process of critically analyzing observable phenomena. At its core, science is a way of deciding what is or is not factual based upon data and evidence. Central to the process of science is reproducibility, documenting what has been done, what has been found, and the conclusions that stem from this. One of the great things about science is that it's true whether or not you believe it, because the data and evidence exists. It has been documented up until this point. So then let's talk about factual versus not factual, facts versus opinions. Facts are statements that can be proven. Now, facts are sometimes misquoted or misstated and can be demonstrated as false or inaccurate, but the take home here is that any fact can be verified. It's something that you can go and Google or hit a textbook or library and find out the correct interpretation of. So some examples. Statistically, women live 7 years longer than men. This is something we can verify by looking at census data or the average age of death amongst women and men in a particular city, state, or country. An example of the false fact: there are 10 inches in a foot. This is false, but we do know that there are inches in a foot, and we could go and verify what the actual conversion is. This is in contrast to opinions. Opinions are statements that are made that cannot be proven. They are often argued. They may be even supported with facts. There may be even some truth to the opinion, like golf is boring. True for a person, maybe, but not for all of humanity. The qualifying attribute is that opinions cannot be reproduced or proven or generalized statements across the board, right? Pizza is delicious. This may be true for a lot of people, but not for everyone. And depending upon who you asked or how you asked it, this may or may not be true or accurate. So the scientific method is a series of steps meant to help us determine what are factual, what we can prove. The method itself is just a series of steps, a protocol or recipe, if you will, for answering questions about the natural world. It's based upon deductive reasoning. Deductive reasoning is a type of logic where we flow from the most general and broad terms to the most specific. Think of it like a letter Sudoku. If we have letters A, B, C, and D in each row and column, not to be repeated, we can use the information in this graphical depiction here to discern and deduce where the remaining B's or C's go. For example, we know that B cannot be in row 2 because it's already present. So B must be in row 4. And that is how deductive reasoning goes. It takes a look at what is currently known, what are the gaps in your information, and how can we fill that information in. So the step 1 of the scientific method, also known formally as the hypothetical deductive method, is to make an observation or ask a question. Science often gets a bad rap for being intimidating, but science occurs whenever someone's curious, whenever someone goes, I wonder why. Why is that? The first scientists, in fact, only sought to explain the natural world around them. Why does it rain? Why do rainbows appear after it rains? Where does the sun go at night? Okay, so they are identifying a situation, asking a question about why this? What next? The next step is to create a hypothesis. A hypothesis is a tentative answer or solution. It is an explanation. Okay? In elementary school, the hypothesis to an experiment might be it's more, it's less. In high school, it might be an educated guess. It's more because, right? In terms of a collegial level hypothesis, what we're throwing out is a potential model. We're throwing out a possible correlation between two items. This causes this because. Basically, what you are defining is what you're looking for, how you're going to know that the effect happened. Okay, these hypotheses are going to reflect past experience, either your own or someone else's. The only criteria for a good hypothesis is that it be something that is observable. You have to see it, taste it, feel it, touch it, hear it, visualize it in some capacity so you know it occurred, and it must be quantifiable. We have to have a way of measuring its instance. How much greater, how much louder, how much more sour is this thing? Okay, so an example, all swans are white. The hypothesis was that swans, by definition, are large white aquatic birds. And the reason for this potential model was that it was testable against the observable world. Anyone who had ever discovered a large white aquatic bird called it a swan. And anytime you talk to someone about a swan, they said, "Oh, you mean a large white aquatic bird? Yeah, I got that." This was tentative, based upon everything we've known up until this point. But then one day, something happened. As the picture demonstrates, there was a large black aquatic bird that hung out with the swans, looked like the swan, acted like a swan. Could it be a swan? This was so surprising that it resulted in the collegial turn of phrase, a black swan event, okay? Something that was unexpected. And herein, we demonstrate how conclusions in science are always tentative. They're based upon the information you had. But when you have more information or a larger subject set, these conclusions may adjust to become more accurate. And so as we looked at the instances of black swans versus white, and we were able to verify through genetics and behavior and reproduction and familial lines, we can see that swans come in two colors, white and black. The white is the dominant form, but they can, in fact, be black. The third step of the experiment, or the scientific method, is to do the experiment itself. Now that you have a hypothesis, now that you have a model that might work, we need to recreate that model in a laboratory setting to verify. Okay? The idea is this can be done in a field or in the lab, but the idea is you want to know when you're starting and when you're ending, so you can take into account as many factors and variables as possible. This leads us to step 4. Once the experiment has happened or has begun, we collect data along the way. We take measurements, numbers, all those observable and quantifiable instances are recorded. And then step 5, we review that data and draw conclusions. How much more? In general, what's the average? We take a look to determine, what does this mean beyond this single experiment? It's creating a relevant framework, a very focused, specific area in which we can explore science and hopefully a single relationship. The experiment itself tries to recreate something that's happening in the wild. Every experiment contains variables. And just like in math, these variables can be anything. Okay? They are something, a thing, event, an amount that can affect the occurrence of the relationship. It's important that you know that variables come in essentially 2 forms, the independent variable and the dependent variable. Know the difference between the two. The independent variable is what's going into the experiment. This exists prior to the start of the experiment. The scientist has chosen these variables. They are arbitrary, and the scientist is manipulating them based upon amount, frequency, or interval in their potential model. The dependent variable, on the other hand, that is a D for data. The dependent variable changes based upon what group it's in or how long the experiment has been going. Okay, so the dependent variable is going to fluctuate and be recorded over the course of the experiment. Controlled variables are a specific type of independent variable, and they get a special name, controlled, because they're consistent throughout the entire experiment. Okay. This is different than controls. Okay. Controls are baselines or comparisons. Any good experiment should have a positive control as well as a negative control to provide context for what change you're seeing in your experiment. The idea behind a positive control is, in this instance, we know for sure something's going to happen. A change will occur. In the negative control, we know for sure that nothing should take place here. This provides us proof that our model, that our entire experimental setup makes sense, that it works. So the example that I have here is an agar dish for bacterial growth. And I wanted to know how dirty my keyboard was in my office. So this is my experimental plate where I swabbed the keys and swabbed the agar and grew up bacteria. If I just looked at the experimental plate, I wouldn't know if that's a lot of bacteria or a little or normal. I need context. I need a baseline, a threshold to which I can compare it to. So in the positive control, in this instance, I poured straight bacteria onto the agar plate to show that if there was a large amount of bacteria present, this is what you could get. Kind of a worst case scenario story here. In the negative control, I have just the agar plate and I did not add anything to it at all. This provides me a comparison to where I could see that the plate itself was not contaminated before even beginning the experiment. This helps us to address how accurate this model was and how believable our data should be moving forward. In terms of the data we record, they basically fall into three categories. Know the three different types. There's nominal, ordinal, and interval ratio. Nominal data sets are those that have a name. In fact, that's what nom de plume means, the name of the day. Okay, so a nominal category has no intrinsic order, no intrinsic value or importance. Okay, so what ethnicity do you belong to? Caucasian, African, Hispanic. These are just categories, and all of them are, in theory, equally likely. What type of pet do you have? Dog, cat, bird. Just different categories. Being a dog owner doesn't potentially make you any better or worse of a person than someone who's a cat person. The ordinal data set, however, is progressive. Okay, there's order in these numbers. So perhaps you're looking at the dosage of a medicine at 1 milliliter, 2 milliliters, 3 milliliters. Okay, and so there's going to be some sort of linear flow here. In terms of the interval ratio data set, these are data that have been pre-calculated beforehand. So if I were to create a table that included the averages of the GPAs of the students in the class, that would be an interval ratio. I've already pre-calculated to create a baseline and comparability, a correction for the variation within our group. Right, so some basic calculations, some of these you're aware of, just to refresh on how to get them. The mean is also known as the average. This answers the in general. To get this number, you take all the data points you have, add them up, and then divide them by the total number of data points. It's finding you the in-between mark. The median is the actual exact middle value. And to get this, you have to take all those data points, order them from small to large, and then work your way out from the extremes to find the exact middle, which may or may not be a whole number. The mode of a data set is the most common, the most typical number. So again, you order them from smallest to large, and you look at which number repeats more and more and more and more. The number that repeats the most, in this case, the 2s and 5s, which were both duplicate numbers, is the mode. And the range explains the overall variety. In this case, you're taking the most large number, subtracting the most small number, and then saying, across the board, this is how disparaging or varied the information could be. In science, we often use the mean and the range. When you're presenting data, there is a particular tool for each job. The right tool can maximize differences and really help your conclusions pop and make them accessible to your reader. So, a couple things to note here. Tables are great for very large data sets that are self-explanatory or for interval ratio information. Okay, so we're looking at this table set example. We can see the 25 brands of American cigarettes and how much nicotine they have. And anyone could look at this and notice that the greatest number, 7 frequency, occurs for 15 milligrams of nicotine. So while nicotine can vary from 3 to 30 times, a 10-time difference in quantity of nicotine, usually it's right about half, 15 milligrams. Okay, so this is a great figure for a table. Line graphs are typically used for ordered data, ordinal data sets, or interval ratios. But generally, they're used for trends over time, or when you're looking at consecutive data sets. So the example here is Sam, who's new to CrossFit training and is trying to bulk up. We can see that in January, Sam's weight is 48 kilograms. By February, he's packed on about 5 kilograms. By March, it's a little bit higher, 6 kilograms, 7 kilograms. The idea here is that line graph connects these data points to tell us that it's one continuous story. And very quickly, we can look at this and go, Sam's getting swole. Bar graphs, on the other hand, depicted in the red, green, blue, and yellow here, are good for group comparisons. And that makes it perfect for nominal data sets where it doesn't really matter if group 1 is red or group 2 is red and group 1 was instead blue, because each group doesn't really matter except in contrast to its neighbor. Okay? So bar graphs are great when we're looking at things like males versus females, the average age in of BMCC students, how many are below 20, how many are 20 to 25, how many are 25 to 30, and so on and so forth. What's your favorite type of breakfast cereal? And if we name them by category type, this would be perfect for a bar graph. Like they say, a picture is worth 1,000 words. So you really want to use the appropriate figure to showcase those differences and tell the story of your data set. Once you've got a figure, we can look at trends and patterns in that data set. One of the common things we look at is the line of best fit. We look at how close those data points are to each other. In this first panel, we can see that all those blue dots seem to point towards an up and to the right line, and they're all close together. This would be a positive correlation, and we'd say it's a tight fit. In the middle panel, they seem to be pointing down and trending downward. So in this case, we've got a negative correlation. As things progress, things get worse. In terms of the last panel on the right, the dots are all equidistant, almost randomly scattered across the thing. In this case, we can't really draw a line. That's the average of this data set. So we say that there's no correlation. In science, we tend to think we need some sort of correlation to answer the question about an experiment. This is not the case. Recognizing that the model doesn't fit is still informative and useful. In terms of lines going up or going down, we can see two examples here, the linear model and the exponential model. In both cases, there's situations where things get better over time or worse over time. The better is solid, the worse is the dotted line. But the idea here is in the linear model, we see an mx plus b standard slope in math. So the story here is that there is a constant rate occurring in this reaction. It's steady, it's predictable. In the exponential model, as the reaction gets started, it's going to pick up the pace, and so the slope is varied. It becomes an asymptote. These are two of the most common relationships, but they're not the only relationships. In fact, there are some 88 different mathematical models that can explain relationships between an X or Y variable or two variables in your study. The reason why math is so integral to science is that by analyzing statistics, we can determine the accuracy and precision of our data. So know the difference between these two terms. Accuracy is how close you are to the true, real value that exists in nature. Precision is a measure of your consistency. So I like to think of this in terms of playing a game of darts. In the first panel, I have low accuracy, but high precision because I've thrown all my darts up and to the right. I'm consistently up and to the right. They're all close together and they're all in the same spot, but I've missed the real mark, the bullseye, which is what I was going for. In the middle panel here, I've got high accuracy because I'm much closer to the bullseye, but I have low precision because now some are up, some are down, some are left, some are right, some are closer than others. There's no telling whether the next dart I throw hits the bullseye exactly or bounces off the wall and hits the person behind me. We repeat science over and over again. We repeat studies with more and more data points, more and more samples, until we get values that show us high accuracy and high precision, like we see in the third panel here, where all the darts consistently hit inside the bullseye and are close together. One of the measures we can use to determine is something real is known as the p-value. And we're looking at a data set and comparing, is this difference significant for the number of samples that we have? Is it enough to say it's real? The p-value is considered significant. It's ever less than 0.05. This is considered a gold standard across the board. And really what it reflects is, what are the chances that this occurred purely by chance, by random dumb luck? And so the idea here is when we give something a significance value, we're saying we invest in this, believe in this, stand behind this with some 96% confidence. Okay, and so the example here is we're looking at the average temperature on the East Coast versus West Coast of a single city. Okay, and the city only spans 10 miles. And so we can see that the temperature does appear to be different. If we look at just the height of each bar, the east side is 2 degrees lower than the west side. But when we look at the error bars, we can see that there is no significance behind those data sets because on any given day, the temperature on the West Coast might just be 6.5 degrees Celsius. 5 degrees different? That's not significant. That's not different enough. On a particularly strange day, the East Coast might be 10 degrees Celsius, while the West Coast might be 6 degrees Celsius. So because they overlap, we would say we have no significance, we have no confidence in this data set to say that our relationship or this correlation we're looking at, maybe the east versus west side, rainfall or rain shadow, that has no bearing on the average temperature or the difference in temperature from one side of the city to the other. Okay, so in general, anytime you see error bars overlap, we have to throw out our hypothesized model and say, this model doesn't answer this question. Either it's related to pure chance or there's another model that we have yet to explore. In science, it's important to identify the difference between correlation and causation. A single experiment might identify correlation, two variables that are somehow connected to each other. But one experiment alone will not prove causation. Correlation does not prove causation. There's a lot of things we have to take into consideration to ensure that one begets the other. Part of that is we have to verify repeatedly that that correlation is real with larger data sets and data sets spanning different times. We also have to verify time order, that the cause occurs before the effect. And we have to take an in-depth look at any other confounding variables that might obscure or better explain that relationship. And so the idea here is the independent variable may cause the dependent variable. We often say X may factor into Y, right? Because the independent variable is your X variable and the dependent variable is your Y variable. But there can be other factors that act on both the X and the Y, bridging them and making them appear as if there's a relationship. A good example of this is a study that involved looking at the number of people who were diagnosed with terminal lung cancer and what they had with them at the time of their admission to the hospital. And they found that all the people of various different ages, ethnicities, genders, from rural and urban environments, all the people had matches in their pockets at the time they were admitted. So correlation would say, hey, there's a trend here that individuals with cancer carry matches. Causation is that inductive leap that says carrying matches causes cancer. And we know from just hearing that statement that that's not accurate, that matches themselves don't cause cancer. There's another factor at play here. Why are you carrying those matches? Is it because you're a smoker and that was a cheaper way of lighting up than a butane lighter, which was not easily accessible at that time? And so in this case, that being a smoker is known as a confounding variable, a confusing variable, because being a smoker is what drew someone to carry matches, and it's also the smoking itself and all the additives in the burning tobacco that led to cancer. Now, science is not the only way of knowing things, not the only way of approaching answers or questions in life. But the reason why science is to date the most useful tool we have in critical thinking and assessment is because it is repeatable. You document exactly what you did and how you did it. And that information is available. Science uses controls. We have checks and balances along the way to make sure that this model works as envisioned. The data is recorded and shared. When you're done with your experiment, not only do you verify your information through statistics to see, yeah, this sounds about right and accurate, but that research has to be peer reviewed by other people in your field who can weigh in on, yes, that sounds right, or I don't know, I found something different. How about you do additional studies or additional calculations so we can compare our data more accurately? And only science allows us to separate out connections, like correlation, versus cause and effect, the causative consequences of one attribute and the other. I included this fun picture because when science works over and over and over again, when we have repeat experiments and processes validated, we come up with scientific theories. Now, this term theory is a little bit of a misnomer because it makes it sound like it's just an idea, but gravity is a theory, and we know that to be true. That whatever goes up will eventually come down. I can watch the sheep jump, it's not going to hover there. I can throw up a ball, it's going to fall. So I prefer thinking about scientific theories as scientific observations. They are true, they are proven, they are predictive based upon past experience and large data sets. The reason why we use the word theory, though, is because science is always looking backward. Science is always reflecting upon the experiment that was done, the data set that was considered. And we don't know where a new study, maybe more participants, or a new study, maybe a year from now, where that's going to land us because technology changes, DNA evolves, there are changes and fluctuations. And so it is both a limitation and a strength of science that our conclusions are only tentative. But every time we redo these things, we are getting more and more accurate. We can trust in science because of that. Now, there have been times where there have been instances of fraud. In 2008, there was a vaccine and autism claim that has been since disputed repeatedly by over 260 independent studies. But the downfall and distrust of the public towards vaccination remains. There has been a fraud attempt on cold fusion. There was also the Great Tazadeh hoax. And in this situation here, a primitive tribe of Filipino inhabitants was discovered in the rural foothills of the Philippine Islands. And they were presented through science as this frozen in time Neanderthal civilization that somehow had remained untouched and unaffected by the industrial changes around them. And it turned out to have been a completely staged ploy and hired actors to drive tourism. So fraud does happen from time to time. Sometimes it's intentional, sometimes it's unrelated to scientific inquiry, but can lead to downstream misunderstandings or claims. But it doesn't happen often. And it's one of those things that because science is founded in fact, it can be verified. Now that may take time, years, but we can go back and determine how often fraud occurs and what is the real story or real relationship here. So in a survey of published papers in 2011, I reviewed over close to 5 million different science papers and followed them in the subsequent years to see how many of them were retracted, which means withdrawn, and how many of those that were withdrawn because of a minor correction or withdrawn because they were proven false. And only 38% of the 788 were proven false. That means out of the total close to 5 million papers from scientific studies in ecology or mosquito anthology or the American Society of Tropical Medicine and Hygiene or Nature or Cell, there was only a less than 2% possibility of fraud in this particular year. So it's not likely. And even if it did happen, we have mechanisms in place to verify that information beyond its initial publishing and correct that miscommunication moving forward. And so the big take home here is we can trust science because we're aware of its limitations and we're constantly looking back at those scientific shortcomings or scientific blind spots to determine, is this still a thing? Is this still relevant? And we will communicate out if it's not.